Welcome to ASM Live at the 115th General Meeting of the American Society for Microbiology. Welcome everybody joining us today. We're going to talk about some important, interesting, and sometimes surprising discoveries in microbiology. We invite everyone in the audience to join us in the conversation. Please just go to the microphone to ask questions. And if you're listening from your computer, please send in questions via Twitter at hashtag ASM Live or through the ASM Facebook page. Today, we're going to have a discussion about silver technology. I'm your host, Stanley Malloy, and I'm talking to Beth Potter from Pennsylvania State University at Erie. Beth, tell us basically what your study involved.
So we are looking at the antibacterial effect of silver Agion technology on our door handles of our campus at the Penn State Erie campus. We've been looking at this for the past several years, so we just completed our 4th year of our study, and we are essentially looking at just the amounts of bacteria from a culture-dependent perspective, and then also trying to identify the bacterial populations that are present on the door handles and how that is changing given the presence of our silver Agion technology.
When was the technology instituted?
So the technology was instituted 4 years ago, so right at the beginning of the fall semester, and so it's expected to last for about 8 years, is what the company predicts. So we're hoping to take our study essentially from the day one, from when they coded it to the 8th year and beyond, just to see if it's still being effective. But no recoding, so it gets applied and then it slowly releases the silver through the time period.
Could you tell us something about the technology in the silver and how it's released?
So the Agion technology is based on zeolite carriers that contain the silver ions, and so it's an ion exchange mechanism. So when you touch the door handle, from the moisture in your hands exchanges with the silver ions within the zeolite carriers, and so then releases the silver ion so that presumably they can kill any of the bacteria that you left behind when you touch the door.
So I expect that the company that developed this technology has done a lot of background research.
Yes, so there are several studies that show that in a laboratory setting that if you take a steel bar that has been coated with this Agion technology, that you'll see a 99.9% decrease in the bacterial numbers. So kind of the twist that we put onto our study is that taking it into kind of a more real world perspective, and the fact that we're looking at is it still being effective when you put it on kind of trivial surfaces like door handles that are definitely there for exchange of bacteria, but again are not the most hospitable environments for bacteria to survive on. So can we still see a decrease, because silver technology is definitely increasing. So essentially silver is going into almost everything and anything that they can put it into. And I think given the concerns that are beginning to arise based on different studies about exposures to us, exposures to environments, are these actually being effective, putting it in all these different places? Do you actually still see the same kind of decrease that you see in the laboratory setting?
And do you?
Well, we do. So again, it's not as controlled as a laboratory setting is, but we can say that our silver-coated door handles do appear to have less bacteria than our non-coated door controls. So it does appear to be effective. We are maintaining less bacteria. But there are definitely still bacteria on those door handles, so we can still culture those. So that leads to very interesting questions about what's being maintained there. So that's why we've gotten into identifying the bacteria that are on the door handles now, to see what kind of changes we could perhaps see. Because as we kind of all know, that if we study bacteria, they can pretty much live anywhere, and this is just a difference. So this might just allow for different bacteria to survive there, which could be new and exciting.
And so far, do you see differences in the types of the untreated door handles versus treated?
So we have completely analyzed data for only one semester. We've just gotten through another semester. And so what it does look like is so far that there is a little bit difference. And I should say again that this is all from a culture-dependent perspective, so that we do tend to see more gram-positive organisms that stick around on the coated door handles. And that does correlate to studies that have been published before, that gram-positives tend to be more silver-resistant given their larger cell walls. And so it appears that the technology is being a little bit more effective against gram-negatives, at least from our simple culture-dependent perspective that we've looked at.
If you talk about door handles, what are the organisms that we should be most afraid of picking up from a door handle?
So I guess, right, you would expect to see a lot of Staphylococcus, and we see a lot of Staphylococcus. I think we would be more concerned if we're looking at more enteric pathogens. So we have been looking at, in our study, we used 3 different plates, basic plates of just a tryptic soy, a MAC, and an MSA to try to get gram-positives and a little bit of gram-negatives as well, trying to get through. So I should say that we don't really see that many a huge amount of any kind of, we've never picked up E. coli or anything like that on our door handles, really. So for the most part, they seem to be relatively good. So nothing that really, we haven't caught anything that really jumps out at you and makes you kind of scared.
Beth, let's step back for a second. And you're at a unique institution. You're at an undergraduate teaching institution. Mm-hmm. So why don't you tell us something about how you do these studies?
So I do teach at Penn State Erie, which again, it just has, in my lab, it's just myself and my students. So no technicians, no graduate students, no postdocs. And so I've been really lucky to have a wonderful group of students that have worked through the past 4 years through this. And so they've put a lot of time and effort into doing this. And so we have 50 door handles that we have in our study, and they sample those door handles once a week for 6 weeks each semester. And so then they're putting onto 3 different plates, and they're isolating, counting, and then isolating out DNA. So it really is a lot of work, and it's giving them a great opportunity to get some basic skills in microbiology and PCR and whatnot. So I feel like at the undergraduate level, having a good solid foundation and some kind of basic techniques is really what I want them, or I think would be really beneficial for them to have as they move on.
Great. We have a question from the audience.
Hi there, very nice work. Actually, I would imagine that as these campaigns to have people be more careful about how they sneeze, for example, cover your mouth while you sneeze, mm-hmm. may actually work against what you're trying to do, because if they sneeze into their hands, there's going to be a lot of mucus on it. Mm-hmm. If they grab the handles, they're going to cover possibly the whatever antibacterial compounds you have there. That's the first question, if you have taken that into consideration. The second one is, I would imagine that most of the microorganisms that we would worry about being transmitted by door handles and such, especially as during the winter season, are viruses. How active is this compound against viruses?
Well, you have 2 good points. One is that the first point about having people be more careful about that, about how they're doing it is, it's hard. I teach a lot, in my first lab that we do in microbiology, I just teach them about hand washing, and you should see how eye-opening of an experience that still is for them. So whether they hear it and whether we still practice a lot of what we hear, I think is still necessarily, or people being as aware of those. And those are kind of, again, parameters that we can't necessarily control for this experiment, which is kind of, again, part of the just looking and seeing what we get out of it. As to the second question about viruses, yes, the silver technology has been shown to be very effective against viruses. It's just that our institution, we don't have as much to do kind of viral work, and those types of things are not really set up for us there. So it's harder for us to look at that in our study, but it is something that it would be very interesting to work on. We just can't really do it at our institution.
If I could just add, you're really supposed to achoo!
Yeah.
Right here. Then you don't touch doorknobs usually. We have another question from the audience.
Thank you, Beth. This is Doris D'Souza, University of Tennessee, Knoxville, and this is related to Dr. Gary's Toranzo's question about viruses. We work a lot with foodborne viruses, including human noroviruses, and a lot of studies have been done to show that human noroviruses are transmitted via touching doorknobs and handles to the environment. If they are, you said, yes, it's effective against viruses. Now, I was wondering whether this is mainly enveloped viruses or non-enveloped viruses that are the enteric viruses belong to as well. If you could share some insights on that, even though you do not do research on it, if there is some information available on those.
So your question is about the viruses and just how effective it is against?
The foodborne enteric viruses.
Oh, then. I honestly don't know specifically about the viral work. There are several studies that have been published about it. And so I've come across several of those in my research literature kind of huntings. But because we're not doing that, I don't necessarily actively look at all of those different studies. So I don't think I'm the best person to give you an answer on that.
Thank you.
We have another question from the audience.
Very fascinating story. Anytime you try to cultivate little creatures from the built environment, it leads you down many different rabbit holes. One of the, I'm Michael Schmidt. You've probably recognized my voice from This Week in Microbiology, and I actually work on the alternative to copper, silver, which is copper. And one of the things that I've learned is that silver doesn't work as well at room temperature. So have you looked at the temperature profile of the effectiveness of this? There's many studies in the literature about the silver Agion technology only working at really warm temperatures, greater than 40 degrees C, and requiring absolute humidities of about 95 to 100%. So have you track the humidity levels, because your results are pretty encouraging for the Agion technology, and I was just wondering what you've learned about humidity and temperature in your studies.
Very good point. Thank you for bringing it up. Honestly, the answer is no. We haven't looked at the temperature and humidity levels. Essentially, we have just put onto the door handles. We've been sampling them from there. They are at least more standardized in that all of our doors are internal doors, so they're not on the outside. They're not exposed to the environmental elements. But that is something that I think now we should definitely maybe start to include within our study, that we should be able to do fairly easily to see if we are seeing any dependence. So we are seeing somewhat of a difference. So maybe we could set up a little of a trial of experiment to see between humidity or temperature-wise if we see anything different. So that is something for us to look into. So thank you very much.
Right.
This is an interesting question, because the idea is when you touch the doorknob, moisture in your hand provides a relative localized humidity, if you would, and also your hand tends to be warm.
Right.
And so it may be that very short-term activity is enough to achieve the result.
That's the intriguing part, and why you always have to be very careful of how you compare and contrast results that are in the literature done under standardized conditions. You're out there in the wild trying to understand. The other question I had is, where are the doors? Are you looking at bathroom doors? Are you looking at doors to your lab? Or where are the doors?
That's a good point. They are spread across 4 different buildings within our campus, and they're all the entrance doors. So we have double door entrance doors to all of our buildings, and again, we're choosing the internal door system so that we kind of don't have to deal with the environmental aspects. And so we have ones going into our science building, into our engineering building, our gymnasium, and then the student union building as well.
One of the interesting things that you probably already know about is last fall at ICAC, there was this big paper that was released by Dr. Gerber's group out of Arizona State. And what he showed is he put some MS2 phage on literally entrance doors, and then he asked the simple question, where did it go? And it went everywhere within a very short period of time, and the most frequent hotspot was the coffee room. So when you touch the door, do I, as an individual who have encountered that doorknob, actually pick up silver so I could literally track the silver through your building?
So we have tried to look at the silver release, but we teamed up with a local company to look at that, and all of our samples came out below detectable levels. So we weren't able, we need something a little bit more refined to do that if possible. So presumably, the answer is probably yes, you are probably picking up, but we can't determine how much that is.
Well, thank you.
Thank you.
We have another question from the audience.
Yes, Janet Rowe from ASM asks, have you looked at the transfer of microbes from handle to hand, vice versa, the number of bacteria that were transferred in either way from the contact?
No, we haven't looked at the transferring about to do a study about how much is transferred in kind of a normal hand touch. We haven't done that, but again, that would be a really great study for us to look into and try to determine to give our results a little bit more perspective. Yes, that would be good.
Beth, one of the things that's interesting about the silver technology is there are other antimicrobial compounds that have been embedded in virtually everything that you can buy that can be plasticized in the world. And there are a lot of concerns that those substances may be enhancing antibiotic resistance in organisms on our skin come in contact with them. What do you think about the silver technology relative to these other types of issues?
With the silver technology, it's kind of, well, one, it's being kind of championed because it has or very little resistance to it, so that's why it's kind of like the alternative to antibiotics. As opposed for looking at kind of aiding antibiotics, it's a hard question. You can kind of find literature that kind of suggests either way. You know, I just ran into an article the other day that suggests that because of the permeability issues that, so silver and how it works to effectively kill the organism is by multiple ways. One is really affecting that cell membrane and really alters permeability, so it can actually enhance some antibiotics because it can get the antibiotic into the bacteria a lot easier. So. But then again, you know, so you could kind of keep on going back again, back and forth about the question of whether it's kind of aiding or perhaps even aiding the antibiotics or again, leading to more resistance. So
It's very interesting because metals were used so much early on as an antimicrobial before we had other antibiotics.
Right.
And now we're coming full circle back into this realm.
Right, yeah, it had been used in ancient Egypt and whatnot to put into silver into their water jugs to keep their water more safe, and then kind of was in and out for a variety of different things, and then really kind of hit kind of its relevance with burn wounds initially, and then antibiotics came around and kind of took the limelight away, and then as antibiotics went down, kind of key paper by Moyer essentially brought it back into kind of the headlines again, and then it essentially has taken off since then. So yeah.
We have another question from the audience.
Hi, I'm Preston Garcia from Castleton State College in Vermont. I have two quick questions. One, we have hand sanitizer dispensers all over our campus, and I was just wondering if you also have that, and if they're in close association to your handles, and how that affects possibly your data. And then secondly, if you could just maybe talk about logistics and maybe price of 50 door handles on campus. We're at a small school too. I think it's great what you're doing. It's really awesome. But how did it come about that you did this project and cost involved in getting the school on board with this project? Thanks.
So the first question about the hand sanitizers is that we do have a few, but very few, that I say that we have spread around our campus. And typically they're much more internal, so they're not close to our doors, or any of our doors that I can recollect have those kind of really nice and handy. And again, they're the entrance doors, so typically, you get a lot of kind of quick traffic in and out of those. And then the second question is that this project got started as actually a nice interdisciplinary project that we have on our campus. So I was actually contacted by a professor who is in the integrated business and engineering program at our school. And so they were doing, as a senior capstone course, they were going to be using this Agion technology as kind of they were going to be marketing it to a hospital. And she approached me so that we could do a kind of internal kind of experiment to show its effectiveness. And so I said, sure, that we could do that. And we started off with one handrail for that study. And then they were going to do it again the next year. And so we decided to get bigger. And so she had paired with, so that IBE program pairs with a local company to essentially facilitate the project. So the Advanced Finishing in Erie essentially coated all of the door handles for us at no cost for us as part of the kind of organization, the connection that they had with this project. So we've been doing this project and we've been supplying our data to those IBE students, and then it has actually branched out to more of a marketing and asking for people's perspective of it. So of how the students feel about the coding being there and those types of things. And they've actually done, I think, one little experiment at a local gas station as well to see how just the general public reacts to having that silver-coated door handle. So it's actually a really great kind of larger project that is going on within our campus. And so we've kind of benefited from that aspect of it.
Great. Thank you very much.
So the silver technology was provided.
It was
then
provided,
The
yes.
Petri dishes and labor.
And those all come from essentially the funds through mainly through our undergraduate research program, grant program we have for our students.
We have another question from the audience.
Great. I'm Kelly Andrews from ASM. Excellent discussion. I actually have a question regarding the impact on the students' health. Since you started the study, have there been a decrease in the number of students getting sick at your school? Or do you know?
So we haven't really been able to relate, or even sort of allowed to relate our studies to the sickness. I did ask the kind of first couple of years, and it actually seemed to be opposite of what we had seen, where I had gotten some. Our spring semester always seems to have fairly low numbers, and that usually is where the higher numbers of nursing visits occur. So that was a little bit off. So, but again, I don't know how well we can really correlate the two of them. So, it's something that is intriguing, and I think our data is kind of, we would like to have it taken for what it is, and then applied to kind of like, okay, what, having those kind of branching questions from that, but not necessarily can we tie that to it.
Okay, thank you.
We have another question for the audience, and while we're waiting for him to walk to the mic, I want to remind everyone listening from your computers, you can send questions at #ASMLive.
Great, thank you. Dave Westenberg from Missouri University of Science and Technology, and I've been working on similar type of work, silver embedded in other types of materials, and it's a great undergraduate research project, and you mentioned that yourself, and I'd like to hear a little bit more about how what the impact is on your students as they experience the opportunity to do real live research, and where do they take this? Do they present it locally? What kind of opportunities do the students have for presenting their research?
Yes, so again, as I mentioned before, this is all done by undergraduates, and I've had I haven't had any problems getting students to work on this project. I think there's definitely a lot of silver technology in what they buy. They see it a lot, so they definitely have an interest in doing the study. And additionally, I should say that I also put a part of this into my micro class that I teach. So in my lab that those students take, they also get to do a portion of it. So they get an aspect of real kind of research into their lab as well. And a lot of students, I get a lot of really great feedback from them. And again, typically that's where my students come from to work on it as undergraduates, because they do like what they got to do. They like that they don't know, and I don't know what they're going to get on their plate. We never know how many bacteria are going to grow on those plates, or if there will be any bacteria on there. So that has been a great perspective. As for my researchers that I have, typically I have about 8 students now working on this project. I started with 4, and we essentially couldn't keep up with the workload demand. So I've increased it to 8. I think I could actually be at 10 by the next semester. But they do get to do all of that sampling at, we have training sessions, which essentially gives the students the training that they need, and then they get to kind of work it on at their own kind of leisure, which works into their schedule. They do present each semester. We have the opportunity to present their research at the local Allegheny branch meeting of the ASM that goes on, so that circulates between different colleges in our area. So every year I've taken my students to that, and so typically we'll break up the project to quantitation and then identification. So we get 2 groups of students that get to present either orally or in a poster format. And then our own institution does have a presentation or undergraduate conference with some of the very local colleges in the area that they also present their research at. So I usually try to have the older students do it in the fall semester, the younger students then who are coming in get to see the older students do it in the fall, and then they get to do it in the spring. So it's really worked out that we have a really great, and again, I've had a great group of students that have been working on this. So they've really taken to really helping out. So once they get older, they help out the others, and they really step up to put a lot of effort into those presentations, which has been really wonderful.
Well, Beth, this has been a great conversation. I think it's going to be really interesting to see how this technology progresses and maybe impacts all of our lives with all the inanimate objects we come exposed to. But also the idea that every undergraduate should get some idea of what science is all about, and you can't do it without some level of research. And
Right.
so it really is refreshing to see more and more students be able to attack problems like this, where they can learn in a different way than just being in the classroom. So
Yes.
thank you so much for joining us. Thank all of you for being here with us at ASM Live at the 2015 American Society for Microbiology meeting in New Orleans. Thank all of you who have been listening online as well. We hope you'll come back and join us again for the next session of ASM Live.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.